The human skin is the outer covering of the body. In humans, it is the largest organ of the integumentary system. The skin has up to 7 layers of ectodermal tissue and guards the underlying muscles, bones, ligaments and internal organs. Human skin is similar to that of most other mammals. Though nearly all human skin is covered with hair follicles, it can appear hairless. There are 2 general types of skin, hairy and glabrous skin, hairless. The adjective cutaneous literally means of the skin, from Latin cutis, skin. Because it interfaces with the environment, skin plays an important immunity role in protecting the body against pathogens and excessive water loss. Its other functions are insulation, temperature regulation, sensation, synthesis of vitamin D, and the protection of vitamin B folates. Severely damaged skin will try to heal by forming scar tissue. This is often discolored and depigmented. In humans, skin pigmentation varies among populations, and skin type can range from dry to oily. Such skin variety provides a rich and diverse habitat for bacteria that number roughly 1,000 species from 19 phyla, present on the human skin. Structure. Skin has mesodermal cells, pigmentation, such as melanin provided by melanocytes, which absorb some of the potentially dangerous ultraviolet radiation, UV, in sunlight. It also contains DNA repair enzymes that help reverse UV damage, such that people lacking the genes for these enzymes suffer high rates of skin cancer. One form predominantly produced by UV light, malignant melanoma, is particularly invasive, causing it to spread quickly, and can often be deadly. Human skin pigmentation varies among populations in a striking manner. This has led to the classification of people, on the basis of skin color. The skin is the largest organ in the human body. For the average adult human, the skin has a surface area of between 1.5 to 2.0 square meters, 16.1 to 21.5 square feet. The thickness of the skin varies considerably over all parts of the body, and between men and women and the young and the old. An example is the skin on the forearm, which is on average 1.3 mm in the male and 1.26 mm in the female. The average square inch, 6.5 square centimeters, of skin holds 650 sweat glands, 20 blood vessels, 60,000 melanocytes, and more than 1,000 nerve endings. The average human skin cell is about 30 micrometers in diameter, but there are variants. A skin cell usually ranges from 25 to 40 micrometers squared, depending on a variety of factors. Skin is composed of 3 primary layers: the epidermis, the dermis, and the hypodermis. Epidermis. Epidermis, AP coming from the Greek meaning over or upon, is the outermost layer of the skin. It forms the waterproof, protective wrap over the body's surface, which also serves as a barrier to infection, and is made up of stratified squamous epithelium with an underlying basal lamina. The epidermis contains no blood vessels, and cells in the deepest layers are nourished almost exclusively by diffused oxygen from the surrounding air, and to a far lesser degree by blood capillaries extending to the outer layers of the dermis. The main type of cells which make up the epidermis are Merkel cells, keratinocytes, with melanocytes, and Langerhans cells also present. The epidermis can be further subdivided into the following strata, beginning with the outermost layer corneum, lucidum, only in palms of hands and bottoms of feet, granulosum, spinaceum, basale. Cells are formed through mitosis at the basal layer. The daughter cells, C-cell division, move up the strata, changing shape and composition as they die due to isolation from their blood source. The cytoplasm is released and the protein keratin is inserted. They eventually reach the corneum and slough off, desquamation. This process is called keratinization. This keratinized layer of skin is responsible for keeping water in the body and keeping other harmful chemicals and pathogens out, making skin a natural barrier to infection. Components. The epidermis contains no blood vessels, and is nourished by diffusion from the dermis. The main type of cells which make up the epidermis are keratinocytes, melanocytes, Langerhans cells, and Merkel cells. The epidermis helps the skin to regulate body temperature. Layers. Epidermis is divided into several layers where cells are formed through mitosis at the innermost layers. They move up the strata, changing shape and composition as they differentiate and become filled with keratin. They eventually reach the top layer called stratum corneum and are sloughed off, or desquamated. This process is called keratinization and takes place within weeks. The outermost layer of the epidermis consists of 25 to 30 layers of dead cells. Sublayers. Epidermis is divided into the following 5 sublayers or strata. Stratum corneum, stratum lucidum, stratum granulosum, stratum spinaceum, stratum germinativum, also called stratum basale. Blood capillaries are found beneath the epidermis, and are linked to an arteriole and a venule. Arterial shunt vessels may bypass the network in ears, the nose, and fingertips. Genes and proteins expressed in the epidermis. About 70% of all human protein-coding genes are expressed in the skin. Almost 500 genes have an elevated pattern of expression in the skin. There are less than 100 genes that are specific for the skin, and these are expressed in the epidermis. An analysis of the corresponding proteins show that these are mainly expressed in keratinocytes and have functions related to squamous differentiation and cornification. Dermis. The dermis is the layer of skin beneath the epidermis that consists of epithelial tissue and cushions the body from stress and strain. The dermis is tightly connected to the epidermis by a basement membrane. It also harbors many nerve endings that provide the sense of touch and heat. It contains the hair follicles, sweat glands, sebaceous glands, apocrine glands, lymphatic vessels, and blood vessels. The blood vessels in the dermis provide nourishment and waste removal from its own cells as well as from the stratum basale of the epidermis. The dermis is structurally divided into 2 areas, a superficial area adjacent to the epidermis, called the papillary region, and a deep thicker area known as the reticular region. Papillary region. The papillary region is composed of loose areolar connective tissue. It is named for its finger-like projections called papillae, that extend toward the epidermis. The papillae provide the dermis with a bumpy surface that interdigitates with the epidermis, strengthening the connection between the 2 layers of skin. In the palms, fingers, soles, and toes, the influence of the papillae projecting into the epidermis forms contours in the skin's surface. These epidermal ridges occur in patterns, see fingerprint, that are genetically and epigenetically determined and are therefore unique to the individual, making it possible to use fingerprints or footprints as a means of identification. Reticular region. The reticular region lies deep in the papillary region and is usually much thicker. It is composed of dense irregular connective tissue, and receives its name from the dense concentration of collagenous, elastic, and reticular fibers that weave throughout it. These protein fibers give the dermis its properties of strength, extensibility, and elasticity. Also located within the reticular region are the roots of the hairs, sebaceous glands, sweat glands, receptors, nails, and blood vessels. Tattoo ink is held in the dermis. Stretch marks, often from pregnancy and obesity, are also located in the dermis. Subcutaneous tissue. The subcutaneous tissue, also hypodermis and subcutis, is not part of the skin, and lies below the dermis of the cutis. Its purpose is to attach the skin to underlying bone and muscle as well as supplying it with blood vessels and nerves. It consists of loose connective tissue, adipose tissue, and elastin. The main cell types are fibroblasts, macrophages, and adipocytes. Subcutaneous tissue contains 50% of body fat. Fat serves as padding and insulation for the body. Development. Skin color. Human skin shows high skin color variety from the darkest brown to the lightest pinkish white hues. Human skin shows higher variation in color than any other single mammalian species and is the result of natural selection. Skin pigmentation in humans evolved to primarily regulate the amount of ultraviolet radiation, UVR, penetrating the skin, controlling its biochemical effects. The actual skin color of different humans is affected by many substances, although the single most important substance determining human skin color is the pigment melanin. Melanin is produced within the skin in cells called melanocytes, and it is the main determinant of the skin color of darker-skinned humans. The skin color of people with light skin is determined mainly by the bluish-white connective tissue under the dermis and by the hemoglobin circulating in the veins of the dermis. The red color underlying the skin becomes more visible, especially in the face, when, as consequence of physical exercise or the stimulation of the nervous system, anger, fear, arterioles dilate. There are at least 5 different pigments that determine the color of the skin. These pigments are present at different levels and places. Melanin, it is brown in color and present in the basal layer of the epidermis. Melanoid, it resembles melanin but is present diffusely throughout the epidermis. Carotene, this pigment is yellow to orange in color. It is present in the stratum corneum and fat cells of dermis and superficial fascia. Hemoglobin, also spelled hemoglobin, it is found in blood and is not a pigment of the skin but develops a purple color. Oxyhemoglobin, it is also found in blood and is not a pigment of the skin. It develops a red color. There is a correlation between the geographic distribution of UV radiation, UVR, and the distribution of indigenous skin pigmentation around the world. Areas that highlight higher amounts of UVR reflect darker-skinned populations, generally located nearer towards the equator. Areas that are far from the tropics and closer to the poles have lower concentration of UVR, which is reflected in lighter-skinned populations. In the same population, it has been observed that adult human females are considerably lighter in skin pigmentation than males. Females need more calcium during pregnancy and lactation, and vitamin D, which is synthesized from sunlight, helps in absorbing calcium. For this reason, it is thought that females may have evolved to have lighter skin in order to help their bodies absorb more calcium. The Fitzpatricks Scale is a numerical classification schema for human skin color developed in 1975 as a way to classify the typical response of different types of skin to ultraviolet, UV, light. I always burns, never tans pale, fair, freckles. 2 usually burns, sometimes tans fair. 3 may burn, usually tans light brown. 4 rarely burns, always tans olive brown. V moderate constitutional pigmentation brown. 6 marked constitutional pigmentation black. Aging. As skin ages, it becomes thinner and more easily damaged. Intensifying this effect is the decreasing ability of skin to heal itself as a person ages. Among other things, skin aging is noted by a decrease in volume and elasticity. There are many internal and external causes to skin aging. For example, aging skin receives less blood flow and lower glandular activity. A validated comprehensive grading scale has categorized the clinical findings of skin aging as laxity, sagging, riddles, wrinkles, and the various facets of photoaging, including erythema, redness, and telangiectasia, dyspigmentation, brown discoloration, solar elastoses, yellowing, keratosis, abnormal growths, and poor texture. Cortisol causes degradation of collagen, accelerating skin aging. Anti-aging supplements are used to treat skin aging. Photoaging. Photoaging has 2 main concerns: an increased risk for skin cancer and the appearance of damaged skin. In younger skin, sun damage will heal faster since the cells in the epidermis have a faster turnover rate, while in the older population the skin becomes thinner and the epidermis turnover rate for cell repair is lower, which may result in the dermis layer being damaged. Functions. Skin performs the following functions. Protection, an anatomical barrier from pathogens and damage between the internal and external environment in bodily defense. Langerhans cells in the skin are part of the adaptive immune system. Perspiration contains lysozyme that break the bonds within the cell walls of bacteria. Sensation, contains a variety of nerve endings that react to heat and cold, touch, pressure, vibration, and tissue injury. See somatosensory system and haptics. Heat regulation, the skin contains a blood supply far greater than its requirements, which allows precise control of energy loss by radiation, convection, and conduction. Dilated blood vessels increase perfusion and heat loss, while constricted vessels greatly reduce cutaneous blood flow and conserve heat. Control of evaporation, the skin provides a relatively dry and semi-impermeable barrier to fluid loss. Loss of this function contributes to the massive fluid loss in burns. Aesthetics and communication, others see our skin and can assess our mood, physical state, and attractiveness. Storage and synthesis, acts as a storage center for lipids and water, as well as a means of synthesis of vitamin D by action of UV on certain parts of the skin. Excretion, sweat contains urea, however its concentration is 1/130 th that of urine, hence excretion by sweating is at most a secondary function to temperature regulation. Absorption, the cells comprising the outermost 0.25–0.40 mm of the skin are almost exclusively supplied by external oxygen, although their contribution to total respiration is negligible. In addition, medicine can be administered through the skin, by ointments, or by means of adhesive patch, such as the nicotine patch or iontophoresis. The skin is an important site of transport in many other organisms. Water resistance, the skin acts as a water-resistant barrier so essential nutrients are not washed out of the body. Skin flora. The human skin is a rich environment for microbes. Around 1,000 species of bacteria from 19 bacterial phyla have been found. Most come from only 4 phyla: Actinobacteria, 51.8%, Firmicutes, 24.4%, Proteobacteria, 16.5%, and Bacteroidetes, 6.3%. Propionibacteria and Staphylococci species were the main species in sebaceous areas. There are 3 main ecological areas: moist, dry, and sebaceous. In moist places on the body, Corynebacteria together with Staphylococci dominate. In dry areas, there is a mixture of species but dominated by B. Proteobacteria and Flavobacteriales. Ecologically, sebaceous areas had greater species richness than moist and dry ones. The areas with least similarity between people and species were the spaces between fingers, the spaces between toes, axillae, and umbilical cord stump. Most similarly were beside the nostril, nares, inside the nostril, and on the back. Reflecting upon the diversity of the human skin, researchers on the human skin microbiome have observed, hairy, moist underarms lie a short distance from smooth dry forearms, but these 2 niches are likely as ecologically dissimilar as rainforests are to deserts. The NIH has launched the Human Microbiome Project to characterize the human microbiota, which includes that on the skin and the role of this microbiome in health and disease. Microorganisms like Staphylococcus epidermidis colonize the skin surface. The density of skin flora depends on region of the skin. The disinfected skin surface gets recolonized from bacteria residing in the deeper areas of the hair follicle, gut, and urogenital openings. Clinical significance. Diseases of the skin include skin infections and skin neoplasms, including skin cancer. Dermatology is the branch of medicine that deals with conditions of the skin. Society and culture. Hygiene and skin care. The skin supports its own ecosystems of microorganisms, including yeasts and bacteria, which cannot be removed by any amount of cleaning. Estimates place the number of individual bacteria on the surface of 1 square inch, 6.5 square cm, of human skin at 50 million, though this figure varies greatly over the average 20 square feet, 1.9 square meters, of human skin. Oily surfaces, such as the face, may contain over 500 million bacteria per square inch, 6.5 square centimeters. Despite these vast quantities, all of the bacteria found on the skin's surface would fit into a volume the size of a pea. In general, the microorganisms keep one another in check and are part of a healthy skin. When the balance is disturbed, there may be an overgrowth and infection, such as when antibiotics kill microbes, resulting in an overgrowth of yeast. The skin is continuous with the inner epithelial lining of the body at the orifices, each of which supports its own complement of microbes. Cosmetics should be used carefully on the skin because these may cause allergic reactions. Each season requires suitable clothing in order to facilitate the evaporation of the sweat. Sunlight, water, and air play an important role in keeping the skin healthy. Oily skin. Oily skin is caused by overactive sebaceous glands, that produce a substance called sebum, a naturally healthy skin lubricant. When the skin produces excessive sebum, it becomes heavy and thick in texture. Oily skin is typified by shininess, blemishes, and pimples. The oily skin type is not necessarily bad, since such skin is less prone to wrinkling, or other signs of aging, because the oil helps to keep needed moisture locked into the epidermis, outermost layer of skin. The negative aspect of the oily skin type is that oily complexions are especially susceptible to clogged pores, blackheads, and buildup of dead skin cells on the surface of the skin. Oily skin can be sallow and rough in texture and tends to have large, clearly visible pores everywhere, except around the eyes and neck. Permeability. Human skin has a low permeability, that is, most foreign substances are unable to penetrate and diffuse through the skin. Skin's outermost layer, the stratum corneum, is an effective barrier to most inorganic nanosized particles. This protects the body from external particles such as toxins by not allowing them to come into contact with internal tissues. However, in some cases it is desirable to allow particles entry to the body through the skin. Potential medical applications of such particle transfer has prompted developments in nanomedicine and biology to increase skin permeability. One application of transcutaneous particle delivery could be to locate and treat cancer. Nanomedical researchers seek to target the epidermis and other layers of active cell division where nanoparticles can interact directly with cells that have lost their growth control mechanisms, cancer cells. Such direct interaction could be used to more accurately diagnose properties of specific tumors or to treat them by delivering drugs with cellular specificity. Nanoparticles. Nanoparticles 40 nm in diameter and smaller have been successful in penetrating the skin. Research confirms that nanoparticles larger than 40 nm do not penetrate the skin past the stratum corneum. Most particles that do penetrate will diffuse through skin cells, but some will travel down hair follicles and reach the dermis layer. The permeability of skin relative to different shapes of nanoparticles has also been studied. Research has shown that spherical particles have a better ability to penetrate the skin compared to oblong, ellipsoidal particles because spheres are symmetric in all three spatial dimensions. One study compared the two shapes and recorded data that showed spherical particles located deep in the epidermis and dermis, whereas ellipsoidal particles were mainly found in the stratum corneum and epidermal layers. Nanorods are used in experiments because of their unique fluorescent properties but have shown mediocre penetration. Nanoparticles of different materials have shown skin's permeability limitations. In many experiments, gold nanoparticles 40 nm in diameter or smaller are used and have shown to penetrate to the epidermis. Titanium oxide, TiO2, zinc oxide, ZNO, and silver nanoparticles are ineffective in penetrating the skin past the stratum corneum. Cadmium selenide, CDSE, quantum dots have proven to penetrate very effectively when they have certain properties. Because CDSE is toxic to living organisms, the particle must be covered in a surface group. An experiment comparing the permeability of quantum dots coated in polyethylene glycol, PEG, pegamine, and carboxylic acid concluded the PEG and pegamine surface groups allowed for the greatest penetration of particles. The carboxylic acid-coated particles did not penetrate past the stratum corneum. Increasing permeability. Scientists previously believed that the skin was an effective barrier to inorganic particles. Damage from mechanical stressors was believed to be the only way to increase its permeability. Recently, however, simpler and more effective methods for increasing skin permeability have been developed. For example, ultraviolet radiation, UVR, has been used to slightly damage the surface of skin, causing a time-dependent defect allowing easier penetration of nanoparticles. The UVR's high energy causes a restructuring of cells, weakening the boundary between the stratum corneum and the epidermal layer. The damage of the skin is typically measured by the transepidermal water loss, TEWL, though it may take 3–5 days for the TEWL to reach its peak value. When the TEWL reaches its highest value, the maximum density of nanoparticles is able to permeate the skin. Studies confirm that UVR-damaged skin significantly increases the permeability. The effects of increased permeability after UVR exposure can lead to an increase in the number of particles that permeate the skin. However, the specific permeability of skin after UVR exposure relative to particles of different sizes and materials has not been determined. Other skin-damaging methods used to increase nanoparticle penetration include tape stripping, skin abrasion, and chemical enhancement. Tape stripping is the process in which tape is applied to skin then lifted to remove the top layer of skin. Skin abrasion is done by shaving the top 5 to 10 micrometers off the surface of the skin. Chemical enhancement is the process in which chemicals such as polyvinylpyrrolidone, PVP, dimethyl sulfoxide, DMSO, and oleic acid are applied to the surface of the skin to increase permeability. Electroporation is the application of short pulses of electric fields on skin and has proven to increase skin permeability. The pulses are high voltage and on the order of milliseconds when applied. Charged molecules penetrate the skin more frequently than neutral molecules after the skin has been exposed to electric field pulses. Results have shown molecules on the order of 100 micrometers to easily permeate electroporate skin. Applications. A large area of interest in nanomedicine is the transdermal patch because of the possibility of a painless application of therapeutic agents with very few side effects. Transdermal patches have been limited to administer a small number of drugs, such as nicotine, because of the limitations in permeability of the skin. Development of techniques that increase skin permeability has led to more drugs that can be applied via transdermal patches and more options for patients. Increasing the permeability of skin allows nanoparticles to penetrate and target cancer cells. Nanoparticles along with multimodal imaging techniques have been used as a way to diagnose cancer non-invasively. Skin with high permeability allowed quantum dots with an antibody attached to the surface for active targeting to successfully penetrate and identify cancerous tumors in mice. Tumor targeting is beneficial because the particles can be excited using fluorescence microscopy and emit light energy and heat that will destroy cancer cells. Sunblock and sunscreen. Sunblock and sunscreen are different important skincare products, though both offer full protection from the sun. Sunblock is opaque and stronger than sunscreen, since it is able to block most of the UV/UVB rays and radiation from the sun, and does not need to be reapplied several times in a day. Titanium dioxide and zinc oxide are two of the important ingredients in sunblock. Sunscreen is more transparent once applied to the skin and also has the ability to protect against UV/UVB rays, although the sunscreen's ingredients have the ability to break down at a faster rate once exposed to sunlight, and some of the radiation is able to penetrate to the skin. In order for sunscreen to be more effective, it is necessary to consistently reapply and use one with a higher sun protection factor. Diet. Vitamin A, also known as retinoids, benefits the skin by normalizing keratinization, downregulating sebum production which contributes to acne, and reversing and treating photodomage, striae, and cellulite. Vitamin D and analogues are used to downregulate the cutaneous immune system and epithelial proliferation while promoting differentiation. Vitamin C is an antioxidant that regulates collagen synthesis, forms barrier lipids, regenerates vitamin E, and provides photoprotection. Vitamin E is a membrane antioxidant that protects against oxidative damage and also provides protection against harmful UV rays. Several scientific studies confirmed that changes in baseline nutritional status affects skin condition. The Mayo Clinic lists foods they state help the skin: yellow, green, and orange fruits and vegetables, fat-free dairy products, whole grain foods, fatty fish, nuts.
Please subscribe and thanks for watching.